So what you need to know is that cell is something that must have three things. Will anybody like to tell me what are three things?
Is it the cell membrane, nucleic acid, and cytoplasm?
Very good. Is this Javeria or Fatima?
Fatma.
Fatima, very good. Nucleic cell membrane, nucleic acid, and cytoplasm. You are very right that these three things are a must for something to be called cell. That's why we have studied that virus is not cellular, because virus is made by two things. One in the middle is nucleic acid, and then is protein code. So, Ms. Javeria, may I ask you that why virus is non-cellular?
Sir, I just joined AS. I haven't done the chapter, so I don't know much about
No
it.
problem.
I'm
Look,
really sorry.
a cell must have these three things, right? Cell must have these three. So, will Maheen Nayyar tell me that why virus is non-cellular?
Sir, because it has circular DNA.
Sorry?
Sir,
Non-cellular.
because virus has a circular DNA and it is non-living.
No, I'm sorry. Look, virus is non-cellular because virus does
It
not.
does not reproduce.
Thank you, Zamin. Because it doesn't have cell membrane and cytoplasm. For every cell, there these three things are a must. Okay, so
So we can also say they do not reproduce by themselves.
again, sorry.
They does not reproduce by themselves.
Yeah. So initially, you're right, very right, but primarily these three things are a must for something to be called cells. I mean, these three things are a must. So your syllabus tells us that why is virus non-cellular? Because it does not have two of these. It doesn't have cell membrane, and virus doesn't have cytoplasm also. For Javeria, since this bio is new for you, a cell has this one is cell membrane, and this space is called cytoplasm. And then is nucleic acid in the middle. So look, we go further. Within cell, there are many structures. We call them organelles, cell organelles. Organelles literally means very small structures, functioning structures. So cell organelles could be the ones which are membrane bound. I mean, they are covered by membrane. You can say membrane covered. And there are some organelles which are membraneless. So the ones which are membrane bound, they are of further two types. Either they are enclosed by two membranes or by single membrane. So the ones which are enclosed by two membranes, their examples are, anyone?
Is it the nucleus, chloroplast, and mitochondria?
Thank you. So nucleus, chloroplast, and mitochondria. She has rightly said because this is their order in terms of size as well. The nucleus is the biggest. And the ones with single membrane, they are like, ER. Don't worry, we'll do them, right? I just throw the terminology first. ER and Golgi body and lysosome. In a minute, we'll recognize them. And the ones without membrane are ribosomes.
Sir, what about ribosomes?
Sorry, Zamin.
Sir, nothing. I was saying about ribosomes. Yeah.
So some are membrane bound, some are membraneless. So membrane bound could be double membrane or single membrane. So let's try to recognize them now. Dear students, if you consider a cell, so Ms. Javeria, may I ask you what is this all space called? Like what am I drawing this with black? Can you help me name this organelle?
Sir, a nucleus, the circle,
Very good.
the
Nucleus.
black one, right?
Yeah, nucleus in the middle. And anyone, this space is called cytoplasm, right?
Yep.
Yes.
Between nucleus and cell membrane, this space is called cytoplasm. I'll turn on my camera in a while. As since I'm eating, so I'm not turning on the camera. So this is cytoplasm. So dear students, inner part of cytoplasm and then is outer part of cytoplasm. This inner part you can call endoplasm. By the way, in Latin words, endoplasm means inner part of cytoplasm. And reticulum means, reticulum is a Latin word. Reticulum, which means sacs, system of sacs, system of bags, system of sacs. And endoplasm means inner cytoplasm, the inner part of cytoplasm. So dear friends, look, here are some sacs, you know, the bags, they are put one above the other. So we call them endoplasmic reticulum, in a short ER. तो बच्चों आपको समझ आ रही है? ER is endoplasmic reticulum. I mean, reticulum means system, reticulum means sacs, the bags. So if they are put, I want to make it short. I don't know how to. I turn on my camera. I want to be visible. Sorry, but why my picture is disappearing? Anyway, so this part is cytoplasm. So the inner part of cytoplasm is ER. Endoplasm. And reticulum means bags. Bags are put one above the other. This is one, then another, then another, then another. Reticulum means sacs. They are put one above the other. When I turn on camera, so the screen gets covered by picture of names, individuals. So dear students, we have with us Aisha, Fatima, Javeria, Bahim. Zamin is not here. In connectivity or something. So बच्चों ये जो है ना, this endoplasmic reticulum could be called as rough or smooth. Rough is one where ribosomes are attached to this, and smooth is which is without ribosomes. So we call it. We are trying to identify, right? So this is called RER and this is called SER, smooth endoplasmic reticulum. So you know, again, ER is basically endoplasmic reticulum. Reticulum means sacs. Endoplasm means inner part of cytoplasm. So endoplasmic reticulum means sacs, the bags put on the inner part of cytoplasm. And when we say RER, this is rough endoplasmic reticulum. RER are the ones which have ribosomes attached to their surface. SER are the ones without ribosomes on their surface. I hope it's easy for you. Now there is yet another organelle. And you can notice that ribosomes are either attached to the ER or ribosome could be freely found. When ribosome is attached to ER, we call it Javariya beta, RER and SER, how do they differ? Maheen,
Sir, one's smooth and one's rough. Sorry, my mic wasn't working.
no problem. So Maheen beta, what is the difference in smooth and rough?
Sir, on smooth endoplasmic reticulum, there are no ribosomes present, and on rough endoplasmic reticulum, the ribosomes are present.
Very good. Then there's another part near the membrane. That's called exoplasm. So there is another system of sacs. Again, you know, these purple bags. So हमारे साथ फातिमा, Aisha, which school are you from? Is this Aisha Nadir?
Assalamu alaikum. No, sir. It's Aisha Asif.
Aisha Asif, okay. So Miss Aisha, can you please tell me that exoplasm means the inner part of cytoplasm or outer part of cytoplasm?
Outer part.
Very good. Exo is outer part. So look, there's another system of bags towards the outer part of cytoplasm. So we could have called it exoplasmic reticulum, but we don't call it exoplasmic reticulum. What do we call it, Miss Fatima?
Is this the Golgi apparatus?
Very good. This one is Golgi body. And Fatima is very right because they are named after the discoverer. The one who discovered this, Mr. Golgi. And there is another thing, Fatima, with the Golgi body, these ones. You know what
These
are
are the cis and trans vesicles.
they? Very
Trans
good.
are
Excellent.
the ones that come before and cis after.
Excellent. I think last you made a bit minor error in last because trans means away. Anyway, but we don't need to know this much. Thus, she's very right that on the either side of Golgi body, on this side is vesicle, on the lower side are also vesicles. So together, vesicles and Golgi body, together they are also called, they are given one more name. Yes,
The Golgi apparatus.
very good. Golgi apparatus. MashaAllah. Sayyid Muhammad Naki has joined us. So this is Golgi apparatus. If anyone finds something tough, please do let me know. We are just recognizing the different organelles. Okay, Zamin, you have internet issues. No problem, beta. I got your message. So look, dear students, so far we saw a nucleus. Remember, nucleus is two membrane structure. Nucleus has double membranes. There are some holes in the middle. But you see, ER is single membrane, Golgi is single membrane. And you know, some of these Golgi vesicles, they may act as lysosomes. Lysosome. We all know lysosome is, lysis means, we had studied this. Lysis literally means breaking, splitting. We had discussed this like electrolysis. When something breaks with the help of light. Electricity. Photolysis is breaking with the help of light. Hydrolysis is breaking with the help of water. So, you know, lysis means breaking and soma literally means body. It's like Latin languages. So lysosome is something which has, inside lysosomes are hydrolytic enzymes. So lysosomes have hydrolytic enzymes. I mean, they've got enzymes which break down, for example, old cell organelles. Yeah, Mr. Naki, what else can they do? Yeah, they can destroy.
Calligraphy and like the old cell stuff, like
Thank
जो
you.
सेल के अंदर डेड ऑर्गेनेलीज होती है.
Excellent. They say cell debris. So, you know, lysosomes destroy many things. So lysosome is just a vesicle that breaks off from Golgi. Our job is just so far to basically identify different organelles. There are two more very important organelles. I better draw one here, this one. So it's called mitochondria. You must have heard of mitochondria that they, excuse me, I received a call. Please just a minute. Assalamu alaikum. Hello? Ji, assalamu alaikum. I'm sorry, I just received call from an elderly. So, Bacho, this is mitochondria, and we know the job of mitochondria is. Allow me to mute the mobile. Okay, so dear friends, mitochondria, will anybody tell me what's the function of mitochondria? The power,
Cellular respiration,
very good,
aerobic respiration.
aerobic respiration. I know who is speaking. Aerobic respiration, yeah. It gives power to the cell. And there are two membranes, outer membrane and then inner membrane. And we can see that inner membrane has folds called cristae. For many of you, it's just a repetition. But I thought that chapter should begin from the very beginning. And there's one more organelle, dear students. We call it like chloroplast. See, this is also two membranes. So far what we saw that a cell may have many organelles. Some are double membrane, some are single membrane. About ribosomes, will anyone tell me if ribosome is single or double membrane?
It is
Ribosomes
single.
are less membraneless.
Wait, I want to see who is responding. Okay, those who believed double membrane, can you raise your hands? If you know how to raise your hand. I think on Zoom there's an option. Those who believe ribosome is covered by single membrane. Oh my God, Mahin, I'm really sorry you're alone. And those who believe ribosome is covered by no membrane. Wow. Javeria and Fatma Sabahat, thank you. Yes, so ribosome has no membrane. It's a membrane less. So let's study a bit more about ribosome. So dear students, this ribosome looking black dots here, then ribosome is, they could be found attached to ER. In that case, we call it RER. So now, something more about ribosome. Please allow me to take this up. There's some very important information. So ribosome has two subunits. Look, its structure looks like this. A bigger part and a smaller part. So what do we call it? Dash subunits.
Bigger and smaller subunits.
Yeah, very good. Two subunits. A bigger and smaller. And but both subunits are made up of two rRNA and protein.
Sir, amino acid as well, or am I wrong?
Yeah, wait. I can't establish the identity. Yes, please. Who was speaking?
Sir, me, Javeria.
Javeria, mashallah. Ribosomes are which join the amino acids, you know?
I think, because like I gave the chapter a read, but like I'm not sure, I
Yes,
might
you're
be
right, you're
wrong.
right. Yes, this is their job. They join amino acid. After this labeling things now, after identification, we'll go on to their functions in the next class, right? This chapter will just end up in
Okay.
3 sessions, inshallah. So, but your 2 subunits, both are made by ribosomal RNA and protein. And so ribosome is also called ribonucleoprotein. Ribonucleoprotein. Why? Because it's made by rRNA and protein. And there are 2 types of ribosomes. One is called ATS and the other is called 70S. And the purpose of ribosome is to make polypeptide. Ribosomes make polypeptide. As Javier says, they combine amino acids and make protein. Its size is about 20 nanometer. Now, so what do lysosomes do? We just saw a bit. So let's talk about functions of other things. Dear students, so what does ribosome do? Hello? They join?
Make
Um,
polypeptides.
They join amino acids to form polypeptides.
Very good. And then, RER, they modify that protein. When ribosome join amino acid to make polypeptide, protein, so RER modifies them. Look, ribosomes are attached to ER, making a rough ER. RER stands for rough ER. Rough endoplasmic reticulum. I better write on here. Rough So it's rough endoplasmic reticulum. So as Javier tells us that ribosome combines amino acids to make polypeptide and initial protein, and that is then modified by RER. This modified protein goes to Golgi body. And Golgi body then modifies it further. Golgi body does not only modify proteins. The job of Golgi body is not only to further modify proteins, but Golgi body modify other substances also. Will Mr. Naki tell me what other substances can be modified by Golgi body?
Lipids
Thank you, Naki. Very good.
and carbohydrates, maybe.
Naki is very right. That basically, I found this. Someone technology friendly boy can tell me that how can I reduce this all outlook? Give it just for Zoom. We're going to finish it up. So anyway, so, okay. So dear students, this is all what ribosome RER will modify this, and then it will send it to Golgi body for further modification, and then Golgi body after modification will store this in the vesicles to be sent outside the cell, inside the cell, wherever they are needed. Remember, lysosome is the Golgi vesicle, but not every Golgi vesicle is lysosome. Lysosomes are those which contain hydrolytic enzymes, but not every vesicle is lysosome. But anyway, so as Lucky says, SCR, remember SCR has a very important role in lipid synthesis. Yeah, you may say lipid-based hormones. So this is the function of SCR. So the basic like lipid-based hormones are estrogen, testosterone, the ones sex hormones. They are lipid-based. They are derived from cholesterol. So SCR actually modifies them. SCR makes them, and Golgi will modify them further. So you can imagine that Golgi is doing very important functions. Let me quickly write on what Golgi does. before we go to the next page. So Golgi body will modify proteins. But is it too much? आप समझ रहे हैं? Golgi body will modify proteins coming from RER. They also modify other substances. Acha, may I know other substances? Golgi body can modify other substances also, like lipids. Will anyone tell me what else is what that Golgi does? A function that begins with S.
Do they store proteins and secrete proteins?
Very good. Storage, packaging, and also secretion. So in a way, they are called factory of the cell. Golgi is actually the factory. Okay, allow me to establish the ID. iPhone. May I know, please, who is this guest? Is it Shahir? Zamin? Anyway. So dear students, this is all what we do for how different organelles function. Let me quickly recap that. Yes, we left one MTOC, microtubule organizing centers. So MTOCs are also membraneless organelles. They organize microtubules. You know, microtubule is like what do you call it? A tubule is a very, very small tube, and microtubule means the ones which, you know, make cilia and flagella. You must have heard of cilia, the hair-like structure that comes out of a cell. Flagella, through which, like, some bacteria have flagella. So MTOC, microtubule organizing center, basically makes so many structures from those very thin tubules. So that's why we call them organizing centers. Anyway, ribosome combines amino acid to make protein. Lysosome has hydrolytic enzymes. RER modifies protein made by ribosome. And SCR makes lipids. Ribosome could be freely found or could be attached to ER. And then Golgi has many jobs. Golgi will further modify protein, will modify other substances, will store them, and will secrete them, or will send them out of the cell if needed out. So there are so many things. Golgi is just like factory. And yes, one more thing about unit conversion, and this chapter is important. So unit conversion. There are just two more topics, right? One is unit conversion. Well, we need to know that 1 millimeter, rather 1 centimeter equals dash millimeter. Anyone? Hello, Bacho?
10.
I really regret that I cannot find who is answering me. I really want to know. Wait, let me do a trick. Yes, okay. 1 centimeter is how many millimeter?
10.
Thank you, Fatma. 10 millimeter. And then 1 millimeter.
सर
Yes.
सर ये मीटिंग 4 मिनट बाद बंद हो जाएगी.
Yeah. So, कोई बात नहीं. 1 millimeter is 1000 micrometer. And बच्चों 1 micrometer is 1000 nanometer. So please quickly help me convert this. Allow me to ask some people. Miss Naki, 5 centimeter is dash micrometer. 5 into 10 is power
10 power 4.
4. Look, Naki is right because centimeter to millimeter is 10, and then millimeter to micro is 10 power 3. It means 1000. So 3 and 1. This 10 means 10 power 1. So this is 4. Okay, Miss Javeria, 5 millimeter. And I want to convert them to nanometer. So I should multiply 5 with what?
Sir, 10 raised to power 3.
No, look, millimeter to nanometer. If you multiply with 1000, it will be micrometer. But for nanometer, you need to multiply with 1000 again.
10 raised to
6. Look, millimeter to micro is 1000, multiply 1000 with 1000. Micro to nano is multiply with 1000 again. Miss Mahin Nayyar, I want to convert 5 micrometer to millimeter. What should I do? 5 into 10 raised power
-3.
-2. Yeah, you divide 5 by 1000. Very good. So you people know this. So there's one more thing that I want to bring to your knowledge. That's prokaryotic versus eukaryotic cell. In fact, there are two types of cells. So prokaryotic cells have no membrane-bound organelles. So, Ms. Javeria, do you think prokaryotes will have mitochondria?
Sir, if it has like, wait, it doesn't have a membrane, but it does have a nucleus, so I think yes.
No. Look, nucleus is a double membrane organelle. Nucleus has 2 membranes. Do you remember that nucleus has 2 membranes? Nucleus, mitochondria, and chloroplast, they have 2 membranes. So prokaryotes don't have nucleus, right? Do you understand my point? Ms. Javeria, prokaryotes do not have, they don't have nucleus, they don't have chloroplast, they don't have mitochondria, they don't have ER, whether smooth or rough. They don't have
सर
like,
लेकिन यूकैरियोट्स में 80s राइबोसोम्स होते हैं और प्रोकैरियोट्स में 70s राइबोसोम्स होते हैं तो
you're
ये
right.
बुक में
Yes,
लिखा हुआ था
right. So yeah, these things are not there. Yes, they do have ribosome because that is membrane less. And one more difference, she's right. Prokaryotes have circular DNA. Circular, but the ends are joined. And they have just one DNA. And circular DNA. And they also have PPG wall. They don't have cellulose wall, right? Their cell wall is PPG. And their ribosome is 70S only. But in eukaryotes, we have all these things plus 80S ribosomes.
Sir, so eukaryote does not have a nucleus?
No, they have only nucleic acid. Right?
So eukaryote does have a nucleus, right?
Yeah, eukaryotes means, eukaryote literally means true nucleus. U means true, karyon is the nucleus. So we'll resume with this next time, and I